Frigid Sea Star, both of which have had their arms up in the water column.
How's that?
Just a quick establishing, yeah, great. All right.
Right. So I bet Sirius is close to stopped. Do you think we can make a move more westerly now?
Yeah, can I center you up and then see what center it is?
Let's do that.
All right, I'm rotating starboard.
Yep, go ahead. Okay.
Let's see.
Down a little gully here. It's hard to say.
Perhaps we can just keep moving up this little gully that we're in right here.
Okay.
1 second, pilot.
But I would say 240 is the direction
Pilot,
of the gully.
if we could please move up this gully.
Can do. We'll call in the next ship move this direction.
Yes, I'm looking at you. 240.
Let's do it. Let's do 40 meters this way.
Copy that. I'm good with that.
Same speed?
Yes. Thank you.
Okay, 40 meters, 240.2 knots. Bridge, this is Nav. Hello, bridge. Could I have another move, please? Range 40 meters, bearing 240, speed 0.2 knots.
So we're about an hour into the dive. We have another, let's see.
Good copy, bridge. Thank you.
30, about 2 hours and 40 minutes left. We are
do you
at
have lasers on?
5,899 meters. So we are coming to you live from the NOAA ship Okeanos Explorer. We are in a place called Hadel Wall at about 5,900 meters in the Marianas Trench, National Marine Monument. We are on the final leg of the 2016 Deepwater expedition of the Marianas. So we're, in fact, this is our final biology, geology dive. Tomorrow's dive will be more cultural heritage, looking at potential sites where B-29 bombers may have crashed during takeoff from landing strips, a landing strip on Saipan. Okay, we're seeing another spiral. So possible evidence of acorn worms.
Watch lead pilot.
Go ahead, pilot.
So this isn't, nothing's very steep around here, but there's this, I've been calling it a gully that had to have been formed some way. Do you know how it was formed? It almost looks like a river.
Yeah, I have no idea. Okay, so our pilot's asking if, you know, because it's fairly flat here, and he's asking how this gully was formed, because it kind of looks like, when you look at it, it kind of looks like a riverbed.
Nav, when was the last move made?
Last move was 3 minutes ago.
Oh, yeah. Copy that.
So we're on our way for 40 meters.
Yes. Probably another 7 or 8 minutes of moving.
No answer on the subsea
No answer
gully?
on that question, Carl. Sorry, and I'm not the one who can answer it. So
It's very
we have
clear.
a couple of questions about whether or not that that light-colored sediment in that
Yeah.
gully is evidence of something biological that's stirring that up, bioturbation. Maybe we could take a quick zoom in on that,
Sure.
please.
Video that's
Real
going on
quick
this
snap zoom on that, so we can get a closer look,
bioturbation.
possibly.
You might be tugging me a little bit, pilot.
Yeah,
Yeah.
I'll stay here.
Not sure. Jeff
I can
Drazen,
come
can
down
you
a
give us
couple
any
meters if you
insights?
want.
Have you seen?
Okay.
Oh, looks like
Holding partial.
pteropod tests down there. Okay, thank you, pilot.
You're welcome. Can come wide video.
Yeah, hi Shirley. I do not know what may have made that little, what looks like a track there. Nope, no clue.
Okay, well, we'll keep moving on up. This expedition is part of a 3-year campaign that was designed to address the Pacific monument science, technology, and ocean needs. Gives the resource managers a better understanding of the deepest parts of our nation's marine monuments. NOAA's Office of Ocean Exploration and Research designed this expedition, and other partners in this campaign include NOAA's National Marine Fisheries Service, the Marine National Monuments Program, the Deep Sea Coral Research and Technology Program, and the National Center for Environmental Information. And through that last partnership, looks like there's something. It looks like it might be another Sea Star up at around 12 o'clock.
I see it.
We'll talk about these partnerships in a minute as soon as we zoom in on what may
10
be
meters
another
to go,
Sea
pilot.
Star.
10 meters.
Thank you.
Yep, another Persingid. Diva Amon says Friella. You can see from the laser dots, which were just turned off, they're 10 centimeters apart, so you can get an idea of scale, how big this animal is.
Okay, video.
We're going to go
Video
ahead
zoom.
and zoom in and just take a look at this so we can document it.
Okay, I'll hold this for just a few seconds.
Okay.
Go in it for. That's max zoom. I'll just
So,
hold this for a few
alright.
seconds.
Those swellings in the arms, not sure what they are, if they're parasites or if that's tissue of the organism itself.
Going out for really quick establishing of the whole organism.
Thank you, pilot.
Good.
Thank you, video.
Thank you, pilot.
So due south is still uphill.
Oh, Holothurian over on the left.
Nav, will you zoom out a little on head pack? Maybe we can see what's going on. No, there shouldn't be
No, that
uphill
should.
due
It's
south.
strange.
Okay, video. Can zoom. And we'll get lasers when we pull out.
Sounds good.
Okay, yeah, we'll get lasers on this when we pull out. So this is a sea cucumber, Holothurian.
This one's very speckled.
And we're thinking, I've never heard of this name, Blagadaria. Is that a typo, or is that what it's called? Okay, and Carl, don't go too far because looks like to the
Okay.
right of
Okay,
the
we got lasers on,
Holothurian,
co-pilot?
there may be
Lasers
a
on?
3-fork colony of something to the right of that. Yeah, to that right of the sea cucumber. There looked
Okay.
like there might be something with 3 forks.
Are you wide?
Ah, okay, I see what you're talking about.
That's
That
cool.
looks like a,
The darker thing?
yeah. Let's see.
Okay, you can go in video.
Okay, pilot, ship has stopped.
Excellent.
Copy that nav. That was a 40-meter move.
Copy that.
Copy.
40
There's
meters.
his shrimp perched on it.
Oh, yeah.
It's kind of cute.
And looks like there's a mysid on the top of this.
Excellent. Is that good for that shot?
That's
Watch
good,
it.
thank you. Yes, that's
Okay.
good, thank you.
You're definitely moving now, co-pilot.
Yeah. Definitely
I'll
now.
still keep trying to stay centered, but
So this is
Kind
a
of awkward.
Xenophia 4
That angle.
that we were just looking at. It's a single-celled animal that
A lot
constructs
of rocks
these
look like they're about to
large
just roll too.
structures.
Just for information,
Uh,
pilots, we've covered 120 meters.
This one was an old
and
degraded
we've
one.
inclined to 20 meters. The 20-meter incline is actually consistent with the contours, so there's nothing too wrong
Okay.
and what? How far to the end point?
from our position here.
Want to snap zoom here, video? It's just
Okay,
a spot.
so yeah,
Thought it was something else.
Good eyes.
Okay.
staying on the
Come
rock.
on.
Yeah, I did too, Carl.
225 meters.
Okay, so we're
So we're a third of the way through.
225.
We
Okay,
are,
let's look
and
at that.
we have-
That's good. We're on track. Upper left, Carl, please. There's just a dark area. I don't know if that's a rock or something else.
Yeah, I
Probably a
think it's the
rock.
edge of a rock.
So we've gone about 100 meters in the last hour.
120. I just
120
read that,
meters
yeah.
in the last hour, so that's good. We're making good progress along the track that we planned out. Still able to zoom in and take a look at some of the highlights that we're interested in getting more documentation on.
Okay, so we still have 2 hours, 2.5 hours on bottom time.
Looks like the same.
Yeah,
yeah,
it looks
is
like
that
the
just
same
current that just
is
like
that is it? Would it be the same one, or are we just in a different area?
Oh no, we've moved on.
Okay, so we've moved on. So it looks like
Wanna
we've
get a
got
quick zoom
a
here, video?
very quick one. Yeah, snap zoom of that.
Although the other ones had 9 arms, I think.
Oh,
and
something on
there's
the bottom, too.
something around.
Something there at the bottom
such
looks like
thing
a
as a quick zoom.
shell. Yeah.
It's max zoom.
Okay. Well, I can go closer and sit down if there's 2 things. If you want to come wide.
Okay, so possibly dead gastropod or snail shell. And then we're just going to do a quick, what we call a snap zoom, just to get some quick documentation,
the
photo
move end?
documentation.
37, so 4
these
minutes ago.
animals that we're
35
seeing.
minutes ago.
Okay.
It's important because even
Pilot,
though
would you be fine if we get
some of
another
these
move
animals may have been dredged up before,
How long ago was the last one?
these data will be made publicly
Started at-
available, so it's
The last
important
move
to
started
have
at
good
29.
documentation
Just
so that
12
the
minutes.
entire scientific
And finished
community
at
has
37,
access
so
to
it's
this
now been finished
information.
5 minutes.
Let's wait. I think in like 1 or 2 minutes, so we don't start stacking them too much.
Yeah, we can go
Copy.
ahead and wanna
Okay.
go ahead and zoom in on
Yeah,
that so we
zoom
can
in.
get.
Yeah. There's lots of stuff down here.
Look at all that little
Little
stuff in there.
community.
Okay, so it looks like these look like gastropods. It's a snail shell. We're not sure if it's dead or alive. And then just up, I'm not sure what that little spherical fuzzball is in there. No idea what that is. Yeah.
Okay, ready to go up to the...
Yeah, ready to go up
arm?
to the sea star.
Okay,
Yeah.
I'll just tilt up.
Yeah, just tilt up. We'll take a quick look at that.
It's not his
Come on,
best
There we
angle.
go.
Later.
That's
That's
okay.
all right.
That's fine.
It's not a bad angle.
Cool.
Okay. So
We have bio stuff all over the seafloor.
watch leads. This
Ye-
is pilot.
Yeah, go ahead, pilot.
So we have proceeded 120 meters, most south, southwest, and the terrain is still really rising straight south of us, which is strange, given the bathymetry we have. So do you just want us to go uphill and follow the local uphill direction, which would be basically south, or do you want us to kind of continue going
So, Carl,
sideways?
can you say that on the Okeanos Explorer line, please?
Oh,
Okay.
sure. So the bathymetry we have shows the wall rising to the west, but we found for the past 120 meters, and as we look ahead with the sonars, that it's rising almost due south, and we've been kind of splitting the difference and going southwest, but it would be, we could go along more easily if we just went due south uphill.
Okay, so folks at home, the shore-based science team, what do you think? Continue going south uphill?
Sure. Yeah, we can
Yes.
keep doing that.
That's affirmative, Carl. Thank you.
Okay. So yeah, co-pilot, after you stop this move, we'll just turn and do the local turn,
Yeah.
which will be a lot easier.
Yeah, that won't be a problem. So yeah, you're about 40 in my camera right now, so
Okay. Yeah, you're still moving on this, right? As stretched out when I started that zoom.
yeah.
All right. Well, as you keep moving, I guess we can start spinning over because you don't have too much farther to go, right?
I wouldn't think so now.
Yeah.
16 minutes total. I would say only a minute or two left.
Yeah.
We've been averaging about 15 minutes between moves, so we're getting about
Okay.
there.
So what will uphill be for you then, co-pilot?
Can you permission to rotate?
Yes.
Get a little better view. Let's see. So this was 220. Let's see what sonar says. So this is roughly 210.
That's better.
Yeah, maybe like 205.
I like it.
Yeah, so you're dead center.
Okay, here we go.
I'm at 205. That looks pretty good for mine.
All right, let's continue that way.
All
Hopefully
right.
it'll start wrapping around westward.
Do you want to do 40 meters?
Yeah,
I don't see much in sonar.
let's do it.
All right, Nav, do you copy?
Bearing 205, range 40 meters, speed 0.2.
Perfect. Thank you.
I'll call it in. Bridge, this is Nav. Hello, Bridge. Could I have another move, please? So we'll have a range of 40 meters, a bearing of 205 and a speed of 0.2 knots, please. Good copy, Bridge. Thank you.
There.
Seeing quite a lot of clusters of the
debris.
leftover from animal stuff.
Possibly, yes.
And you see where it looks like it's coming out of a tube on the left side, on the other side of that slight slope.
I do. Well,
Seen any
quite
more
possible.
pink rocks?
I've not. No, I have not.
We
Well,
saw
there's one
another
right.
one while you
Actually,
were
there's
out at
a.
lunch.
Did you?
Yeah, it was bright pink. I mean,
Really?
It was like a rose, dusty rose color.
Did it have pores?
Did not appear to. What Scott France suggested that it might be a red algae that had fallen down the slope from much further up. Possibly. But he was going to check on a paper that he was looking at earlier and come back with a comment later. Whoa, drop off. There are some pretty impressive little drop-offs in the side of this slope that we're on. This is about a 45-degree slope that we're looking at on the right side of the screen right now. And that's the typical, what we call angle of repose for debris on the Earth's surface. When it's just collecting in a pile, you can make a pile of stuff that will have a maximum angle of about 45 degrees before things start sliding down.
Yeah, there's not much alive down here.
No.
Well, that you can see.
Right. Pilot says there's not much alive down here that you can see.
That you can see.
And he did emphasize that you can see, because most of it is living under the seafloor. Microbes, huge biomass on this planet. It's interesting that as we look across the screen from right to left, we're going into, on the left side, a sequence that looks like it's a mass of debris of enclosed rock fragments with sediment that's a slightly lighter color than below on the right-hand side. And all of it, as we're looking now, and our heading is more like 250 degrees, close to due west, we see a slope that is a little steeper than 45 degrees. That suggests that some of this sediment is a bit more indurated or hardened, maybe cemented, and therefore can make a more high angle slope. If you go to the Grand Canyon, you'll see the limestone cliffs that are vertical, and between them are sequences of rock called shale.
It's just below the camera.
Fish below the camera. Pilot, do you see a fish below the camera?
Look down.
No,
If
but
you
I
look
can go
down.
back.
Okay, we're backing up, Jeff. Was it large?
Did not see.
No, it wasn't terribly large. It was a, typical looked almost like a cusk eel. It just barely crossed the lower boundary of the main camera.
Oh,
And it was
okay.
going to the right and below the vehicle.
Do you see it at all in the Sirius view? I wonder if we can get closer with Sirius? Without tangling up the cable?
No, but I
Yeah.
can't go much further back now.
Right.
Yeah, I don't see.
It may be gone. That's okay. Thanks for trying, guys. It was one of those fleeting observations.
Thank you, Jeff.
Yeah, I don't see anything.
We don't see anything in the manipulator cams or
That's it.
Sirius.
Just straight ahead on the bottom.
Straight ahead on the bottom. Do you see it on the bottom? Straight ahead, he said. His feet's a little slower.
Tilt down a little bit. Tilt down just a bit.
Tilt down just a little bit, he's suggesting. Do you see anything, Jeff? I'm looking at Sirius as
Yeah,
well.
it was there. I think if we back up a little bit, it was heading down slope to the left.
Can we back up at all? Pilot, he was heading downslope to the left, says Jeff.
Oh, I see him now.
Ah, the pilot
You
sees
see it
him.
now?
Sees him.
Right there.
Yep.
Yes.
Yep.
Coming right into the middle of the camera.
Okay, there we
Okay,
go.
can
All
hold
right.
there.
Yes, that's it.
Holding partial. Wow.
And we need to get a little closer without scaring him. Good eyes, Jeff. But somehow
wanna zoom
that's
in?
not
Sure.
surprising to me.
Going slowly.
I'm still far away, but
Taking a bit more zoom.
Okay, I think this is, I'd like to see a bit more lateral view, but this guy looks like a cusk eel called Basocetus. I've seen this guy before, and it's really common in our baited lander work. This species will aggregate in numbers sometimes of almost a dozen, and it usually hangs out around the bait and eats all of the amphipods that are attracted to it. So you can see here it's got extremely well-developed nostrils, which maybe it's using to detect bait, not to come in to eat the bait, but to eat the other animals.
Come out just a bit.
And yeah, I'm hoping this guy might turn a little bit for us. Looks like he might. And we can take a look at and look at the pelvic fins maybe if he's going to let us, but he might not.
Watch lead pilot.
go ahead.
If we can look at pelvic fins, that would be helpful if this thing
Roger.
cooperates at all.
Okay, pilot.
How's the view,
Chipper
Jeff?
stopped.
Copy that, Nav.
Oh, there we go.
He's turning over for you.
That's great video. Thank you very much.
Okay, let's let him go.
Many thanks from the shore, pilot.
Full way.
That was
Nice.
it's amazing. We couldn't see it the first 3 times here.
Yeah, I did not. Yeah, even when you said you saw it, it took me a few moments to.
Well, we were at
find it.
5880,
So I hit 6.5
5880
my camera
meters
pilot, but
Okay.
for that.
we're not moving
Cusk
too
eel.
quick. And
Yeah.
It's not the deepest
Start
fish have
going
been
now. We're
sound.
going
No,
205.
it's not.
yes, I believe that's
And
correct.
actually,
Not sure.
Jeff
Copy that.
was on a cruise on the Schmidt Ocean Institute research vessel, the Falcor, in 2014. I happened to be lucky enough to be out there with them.
Where was that cruise?
That actually was in the Mariana Trench in the second deepest point in the world's oceans, in the Serena Deep.
And what kind of
10,800 and some odd meters.
what kind of technology were you using for exploration there?
What the folks were using were baited landers. These are autonomous landing devices that have a wonderful
looks like a little
camera system
roadway.
on them.
Yeah.
They're
On the
dropped
side
off
of
the back of the ship, settled to the
Cliffs they have
seafloor
in Colorado.
with bait on a long
That-
pole, and they stay there for
So
hours.
it looks like uphill's
And
a
it
little bit
attracts
more
It
right now.
invertebrates and
For you
anything, any
riders.
scavengers, And
brightness is a little bit
Right.
too
fishes
strong.
as well. And then what kind of lights do they have? Kind of
yeah.
reddish lights, or
Yeah.
do they, as soon as they detect motion, the light, the
When
That's
does
a
camera
good
that
takes,
question.
start
I'm
now?
or are
not
they
sure.
continuously
We'll have to
recording?
ask Jeff.
So 9 minutes ago.
Okay.
It would be good for our listeners
They
and
do-
people who are
they do
watching.
continuously
Yeah, go ahead, Jeff.
record.
Go ahead, Jeff.
Okay, I was just going to say, yeah, it's just regular white light. It's at fairly low intensity, but it's at these depths. Many of the animals don't seem to be disturbed at all by the lights, and so it doesn't seem to have much of an effect. There's a thought that at these depths, the animals probably tuned to see bioluminescent flashes. Their normal visual field is rather dark, and when they have these bright white lights, that may actually blind them briefly, but given that their normal light field is darkness all of the time, they see a bright flash, and then that's it. And I will stop talking because we have an awesome...
Yeah, what was that? Right, okay, good. I was just... What we were looking at right there was a comb jelly, or a ctenophore.
Try again there.
Roger.
Okay, go for it.
Okay, we're going to get another view of it, I think.
It was interesting that it pulled in its tentacles on screen, and now it's
Coming in.
inflating its
Yeah, it's a
bell
bit
a
too
little,
close to
moving
focus.
in the water column.
Yeah, it's right in front of you. You're full wide.
Very,
Okay.
very close to the ROV.
Came in quickly.
Did.
We have a lot more rocks here. Very angular, many of them. Most of them cobbled to boulder-sized, large rocks, smaller white fragments.
I think
Going towards my port pilot.
Yeah, I was just going to look over this edge.
Yeah. Do you want me to rotate it all to keep you in
No,
view?
that's okay.
Copy.
we have quite a few scientists participating in this dive today from shore, along with all of you at home who are listening and watching to this exploration of this abyssohedral transition zone. I am
That white
not
thing?
sure. No, that looks like it's right next to a spiral. So let's go
turn
take a
Oh,
look
you're
at
right,
it.
now?
Shirley. Oh,
Let's
can
go
we
take
zoom
a
on
look
this?
at it.
Is it an acorn worm?
I don't know. I don't know.
It
I'm
is right
sorry,
next
Pat,
to a
what'd you say?
spiral.
It is next to a
It
spiral.
is
Turn to
part
follow me now.
of the spiral.
Copy
I'm
that.
going to zoom over here.
Oh, it's an acorn worm.
All right.
Or some sort of a
that's
worm, like an acorn worm. It doesn't have a
Right.
pink head like many of them we saw earlier did.
Pretty much due south. 180.
Wow, this is very cool. And now, I agree with you. The spirals were not imaginary.
Keep lasers off, or?
You can put them on right now.
Yeah, let's definitely get a nice zoom on this.
All right. Right on
This
there.
is at 5,876,
Let's go in partial.
5876 meters.
Cut the whole thing with lasers, and then we can turn
Great.
them off for the rest.
Oh, it's a worm.
Okay,
It is an
take 'em off.
acorn-type worm. It does
Lasers
have
off.
a broad
It's a
head.
very
This is a
beautiful
group
worm.
called
What do you want first, video, the
Enterobnus.
whole thing?
Enterobnus.
And what you are seeing is it in its
is nice.
digestive process.
Oh, you sure are.
You can
Where
see
they
what
go
it's
in a
eating and what
circular
it has
spiral
eaten.
pattern.
Yeah,
and
why
what it
does
has
it
left
have
That's a
to-
behind.
line.
Did, I'll go in on the head first.
And
Okay,
it
you done with this
seems
full body
seems
shot?
to like
That's good.
this
Okay.
fuzzy
Rotate
surface that
this up.
people have been mentioning.
I'll take a partial.
Scott, you want to tell us about this
Acorn worms.
animal?
Tilt up just a bit more.
We haven't heard from you today. We
Perfect.
miss
I'll
you.
just-
Oh, we have. We have heard
Oh,
from
you-
Scott.
For now.
We have. Oh, he called in.
Yep, you
Oh,
did.
I must have been at lunch.
You were. Yeah, it's okay. We'll forgive you. It's late where Scott is. And where is Scott?
He's in Louisiana,
He is in
Lafayette,
Louisiana. That's right.
Okay, I'm gonna take
broiling
a
hot
closer shot
Louisiana.
on the head.
Right,
Okay.
but he could be making his dinner.
Oh wow!
Go right.
We are
Gosh.
looking at the
All right,
head
I gotta Google this.
of this worm. I'm trying to see if I can see
Diva.
him munching. Scott is on the chat. He says it's an ent.
Yeah, Entrepneust. I'm just
Nothing like the ents in The Lord of the Rings.
Okay. There we go. Yeah.
Perfect, yeah.
They are a class of invertebrates consisting of one order of the same name, closely related to echinoderms.
All right, if you wanted to, we could pan down to the end of the worm and then call it good.
Okay. To the
Then
other
we have the
end?
full body, yep.
Okay.
I'll come out just a little bit.
It's interesting that it separates there.
Yes, it is.
Okay.
I think what I remember Scott telling us on an earlier dive is that these two flanges that are wrapped around the upper part of the worm behind the head are, I think he said this was gonadal tissue.
I'm looking right now.
Okay.
And you can see the patch of fluffy
Alright.
sediment that surrounds the spiral, and the area of the spiral has been wiped clean
set.
of that.
Yep, clear.
So the guess earlier
you.
that maybe this might be some microbial material certainly sounds reasonable.
Yep.
Okay, co-pilot, I'll get us centered
Well, Amanda
back on
Nedburn-
205.
Excuse
We're
me.
probably ready to go again.
Yeah, copy that. I think I'm pretty good.
Excuse me, had a coughing fit.
I will
Amanda
rotate
Nedburn
with
explained
you.
on one of the midwater dives about the
Starting
particles
now.
that drop down into the deep ocean. So, you know, the phytoplankton, zooplankton, and the upper layers die, and they start getting digested
something
by
wrong.
bacteria in
Right.
the water column.
Fly by.
And
Yeah. I'm pretty much
eventually,
almost
when you
centered
get enough
anyway.
bacteria, they have this response called quorum sensing, and they
Wanna do
start
this tap zoom there,
to
video?
Sure.
produce
Just do a quick
kind of
snap.
a
It looks very
mucus
similar
and
to
some.
the other things we've-
It's called a, what did she call it? Something ESP, proteinaceous
Is it
substance, some EPS, I think, something extracellular proteinaceous substance, I think is what she said. And they use that to survive, but eventually, by the time
pilots
that those
at
particles
205
make
right
it to
now,
the
so
bottom, there's very little
spot.
left
How long
of
ago was that called in?
nutritional
Just a while
value.
ago, right?
So that's
48,
part of the
so
reason
yes.
why
Okay.
you
So I
don't
think
see
Pierce has actually put a stop
much
sign.
life in the
Yeah,
deep sea, but you do see life. So
I think the
it's,
same
it's
thing
not
would be fine. 40 meters,
devoid of
205,
living
42 knots.
organisms, as was thought.
Do you think
Quite
you
a
could
while
turn
ago.
starboard now?
And
Oh, absolutely.
To look at this.
Just the brightness is more like looks like 10 degrees to starboard or so.
10 degrees. I'll go to, let's see.
yeah, yeah.
So that's 210.
All right, that looks perfect.
210.
It's kind of fussy though. I'm just trying to make sure we go more west.
Yeah, my, I'm oscillating.
Okay, we have a field
5
of
degrees
boulders
both ways,
we're
too.
going over right now. A slight
What?
Five or 210?
rise that
Now,
then is falling away a
that's
bit
215.
to the right-hand side of the screen. There are some
250.
boulders that have black
I think, yeah, even,
215.
upper portions
Yeah.
and
40
a
meters,
white
same speed.
crust on the base.
I think that's good.
Copy that.
I think that'll get us wherever we want to go.
Bridge, this is Nav. Another move for you. Range 40 meters. Bearing 215.
So we
Speed
have
0.2.
scientists participating from all around the world.
Good copy,
We
Bridge. Thank
really
you.
appreciate their expertise. They are calling in sometimes. They are entering data in our chat room, which is very much appreciated. And I'm going to, oh, looks like something pink there too. This is
Oh,
really
yeah, it
interesting.
sure does.
Yeah, let's take a look
This
at that.
is really curious.
What the heck is that?
Well, I think the white material that we've been looking at is carbonate,
Okay, video, you can go in.
I suspect.
Scott France said that he, we were talking earlier about
Wow.
whether or not this could be, this is the same as what we saw earlier.
Yep.
Someone asked if this could be red algae. I mean, obviously down
Yeah, Scott
here.
mentioned that.
Yeah, down here, of course, they're not. It won't be
yeah.
alive. However,
Could the color
yes,
persist?
it could
It is
persist.
clear.
So
We
they're
have
dried ones in the
version of
lab.
science wants a closer look.
Yeah, let's go
this-
in tight on it.
Crustose coralline algae. I'm not sure that's what
Got it.
this is, but could be pink. I'm just not sure what we're seeing here.
That's the thing. Every time we dive with an
Okay,
ROV
go
or
wide.
in a submersible and get down to the bottom of the ocean,
We see things that
We
we've
s-
never seen before.
yeah, we
I mean,
put our
yeah.
eyes on something that no person has ever seen before.
Right.
Nobody has looked at this spot on the seafloor.
And there's so many others. I mean, we select
Oh, yeah.
these few tracks that we can select, but there's so much left.
Well, 70%
who knows?
of our
Yeah.
globe is under the water, and we
And
know
most of that is
every
unexplored.
day we say the same thing. We know that we know more about the surface of the moon, Venus, Mars, and now
Jupiter.
the clouds of Jupiter than we know about the surface of our own planet.
And then there's that huge biome in the midwater column.
Oh, yeah.
That has been,
Think of the volume.
has most of the biomass in the world and really very little. We made some interesting deep water, midwater dives on this leg of our expedition. Seen some animals that have stumped the experts. I think you might, those of you at home may be able to see some of these images on the Ocean Explorer website. Each day our video team selects some, the best of the best. I think all of their images are outstanding. These are award-winning videographers that we have out here. And they select the best of the best, and they're very picky about what they want to
No, it has to be to
display as the
contours
best of the
getting your
best.
dive
They're
in.
not
Is it
happy with
still
it being
30 over 10?
99.9% perfect. I'm looking at Roland right now. Has to be
Might hit 15
100%
degrees, but,
perfect.
Yes,
And
we
and
scan
that's what
through
you're
all
getting.
this, and we scan through
we
all
just hit the 2 hour
videos,
mark.
They do.
2 hours
Yeah.
left on the bottom.
Yeah.
pick out the best.
Yeah,
Making good
hours.
progress.
I
Yes, we
mean,
are.
you know, we're down here today for 3.5 hours, but they're also scanning through videos that they've taken from the time the ROV left the surface and got down to the bottom. So, you know, every day, 8 to 10 hours worth of videos that they're scanning through to select the very best images that are going in the highlights.
Pardon me, there's something on the left bottom of the field of view that's moving. It looks like it's settling towards the sediment. It's
Okay.
approaching the
Okay.
middle bottom of the
Pilot,
image.
we're getting a
Okay.
suggestion from the shore that there's something moving on the bottom.
Bottom of the
Pilot,
image.
we're getting
Okay.
a suggestion from the shore that there's something moving on the bottom of the image settling.
Yeah, I saw
Do you
that.
see it?
Yep.
Okay, let's take a look
It's
at that.
almost directly below the lasers now and whitish.
Okay, thank
Okay, video,
you.
can go in.
Yeah, I think I see right now it's above.
Oh, yeah,
and it's
another
a
fish.
Okay.
It's
Wow,
a fish. Good
Jeff.
eyes. Jeff, you are the...
Well, he is the fish man.
I know.
Okay, take a bit more.
Okay, I'm still far
I
away.
can't claim spotting that one. Chris Kelly got that one.
Ha! Okay. So Jeff is in the Expedition Command Center on the
Exploration.
University of Hawaii campus at Manoa. So tell us, Jeff, what are we seeing?
Okay, so yeah, this is another cuskeel. This is a very common family of fishes at these depths. And judging on the sort of a bit concave head, the really prominent ridges, very tiny eyes, this looks like it might be in the genus Panopus,
Okay.
which is a not very well. Many of these cusk eels are not very well known. We may have seen this one once before, but I'm not certain it's the same species. It may just be in the same genus. I'm not certain. Maybe if Bruce Mundy or Ken Sulak are listening in, they can contribute something here.
We have seen this fish before on these dives, but I don't recall
beautiful.
at this depth.
All right, so do we need to get close to the head and try and get the top of the head and the
gill structure.
gills and the
Go for it.
mouths? Okay, here we go. Here we go.
Coming down.
Tough to focus
It
on
is.
moving critters.
Taurus Smithy's doing a bang-up job, though. So is Carl. So are the entire ROV crew trying to keep this,
There we are.
keep D2. You can look and see on the
Yes,
from the view from the Sirius vehicle how they're maneuvering D2 so that we can keep the
Oh, you can
fish.
see the gill structure in this image. The pectoral fins. These are all diagnostic characteristics of the fish species, so it helps the experts to be able to determine what
Right.
the
saying the head,
genus
the
and
eye, the
species.
gills, the scales on the head, pectoral fins. Yep.
That video is fantastic. Thank you, pilots. Thank you, videographers.
Awesome. All right.
Thanks,
Should we
pilot.
go?
Great.
Just let him swim out.
Oh, now he doesn't want to swim.
Right, now he's going to cooperate.
Yeah.
Okay,
Nice,
great.
Carl.
Oh my.
It's a little.
That's
rack.
I've hit 6-0 on my cam, but
More spiral
moving
trails.
slow, so you're plenty safe.
All right.
Nothing dangerous on sonar. Deck bucket still looks good.
Ooh,
So
lots
Nav,
of
when
them.
was the? Is it 107? Was that when the move was put in?
Yes.
Okay, copy.
Now there are patches where the sediment
3
has
meters
been
to go.
grazed on all over the place. Now,
Good copy.
in that
Could be a
patch up at the
holothurian.
top of the screen,
Yep.
is there a critter in there?
I
See a rock in the middle.
don't think so. There's a rock.
Yeah. Well, there sure was a critter there.
Yep.
It's not spiral trail. It's
No,
all
something
over the
else.
place.
Scott was saying earlier that trails like this in shallower water could be caused by heart urchins. Yeah. Maybe
The
yeah,
animal may be up to the left
up to the left. Yeah.
in that. Pilot maybe.
Yeah,
to
he's
the
going to it.
top
Yeah.
above the rock and to the right of
Okay.
the two small rocks. Maybe the critter is in there. Boy, this guy sure has taken an erratic path. I don't know.
I don't see a critter.
You do or you don't?
No, I don't.
No, I don't either.
I don't either.
Yeah, in fact, you're right. Tara Luke says, looks like that first place that Deb
Triple
asked
stoked.
about the bioturbation, the traces do. Indeed, they do.
Thank you, Nav. Yeah, we could probably rotate
Oh,
to
beautiful spiral.
maybe
These,
225.
I'm just loving these.
Okay.
It looks like center is adjusting a little more starboard.
We
rotate
have
if you
our
want.
shore-based team, Asako Matsumoto from the Chiba Institute of Technology in
Good
Japan.
rotate.
We have a few people
Sure,
from Hawaii,
start that
University of
now.
Hawaii.
So that
Astrid Leitner, Chris
10 degrees
Kelly, Jeff Drazen, Mackenzie Geringer. Who else from Hawaii? I think that's all from Hawaii. We have Debbie Blaney from the Office of Ocean Exploration and Research in Silver Spring, Maryland. Deb Glickson,
Yeah, I think
Nolan
that looks good in
Barrett, and Jorge Monroy from
Harbor Branch.
Now,
Harbor
what's,
Branch at the Exploration Command Center
summer.
there.
Is it pretty much due west to
Oh,
get
sorry,
to dive
Diva.
in?
I left Diva out.
Oh, yeah.
Better
Diva's
not.
from Hawaii.
From the
Diva
still on the
and
ship
Deb
list.
were science
due
leads
think that was
during
just
Leg
random.
1.
Leg 1.
Yeah.
Gene Ranke from the University of Kansas.
Big-
Oh, Hong
pretty
Peng
good
Tong.
spot right
Sorry,
now.
I forgot you as well from Hawaii. We've got quite a large contingent from Hawaii. We have Mike Vecchione from the Smithsonian, Bob Carney from Louisiana State University, Scott France from their arch rival, University of Louisiana at Lafayette, Tara Harmaluk from Stockton University in southern New Jersey, and Tina Molitsova from the P.P. Shirshov Institute of Oceanology in Moscow. Thank you all very, very much for participating in this and just about every other dive we've had on this leg of our expedition, and really enhancing the quality of
Is
the
there still that
information we can
drop-off?
provide to
kinda
our
following.
shore-based
How far is that?
viewers
10
and listeners,
10
as well
meters
as
ahead.
You
to
can kinda see
the
it a little
data
bit.
for this leg of this expedition. So thanks very much.
far
Patty
away.
and I greatly appreciate your participation and
I
partnership.
guess it's more over here.
Yeah, you're going off
The other side of that. Yeah.
starboard. Yeah, right there.
Yeah, we have a few of what look like ripple marks on the surface of the sediment, more or less parallel to the
You're just
slope.
out of my. Yeah, you're coming back.
So I just learned a new word.
Oh,
The
you
word for
did?
the day is neoichnology. So Gene Ranke from Kansas, professor of geology there, said that he's sending pictures of some of these trace fossils, which he explains later, to Dr. Steve Hasiotis, who's an expert on trace fossils at University of Kansas. But then he goes, oops, they're not fossils yet, and they are called neoichnology. So these little spirals and the things that we're seeing on the surface of the sediment.
Mm-hmm.
Hope I got that right, Gene.
What I thought was interesting was that earlier some of the spirals that we were looking at were flattened,
Right.
as if the sediment
Yeah, I don't
that they
see
were
anything.
composed of
Just
had started to
sweeps
spread
in
out a little
from
bit
my
on the
view.
seafloor, and that
Yep.
eventually will be
Yep.
removed as traces by currents, perhaps.
Yeah, looks like we're coming up now onto maybe, I don't know if it's the light that's deceiving me, but it
No,
looks
it
like it
looks
might
like
be
a
cutting
little
bit,
rise.
yeah, a little bit of a rise here.
Yeah.
We are at 5,865 meters. And it looks like there's
To
something
the
on
right,
that
there's
rock.
a very interesting anemone or something to the right, just going out the
Yep.
field of view camera.
Okay.
That is right.
Okay.
But
But you're
it
talking.
might be a sea star.
No, I think he's.
on the right.
Jeff, are you talking
Yeah, just
about
on
down
the-
now
No,
towards the
right
bottom?
there.
Oh,
Oh,
there we
wow.
go. There we go.
There we are. Yep, that
Is that
is.
It's a jelly.
Oh, it's a
Yep.
jelly.
You're right.
Wow.
Wow.
That's pretty cool.
So what we will do, mosey over there and patiently wait for the zoom.
Okay, you want a zoom video?
Here it is.
Oh boy, oh, look at that. So,
That's
what
nice.
is it doing? Look at that.
It's pulsing. That's what it's
pulsing.
doing.
Take a bit
Looks
more
like it's
zoom.
doing something with its tentacles.
It does, yeah. But it looks as if we're looking
That.
at the bell at the
We sure,
top.
yeah, we definitely are.
Mhm. So it's just slowly moving across the sea floor. Anybody recognize this jellyfish?
I'm going to come out a bit, try to get, it's moving kind of fast for the zooms.
Yes, its tentacles are sweeping the sediment surface of the seafloor.
Come in just for one last shot on as it comes through.
Okay. He's coming.
Maybe. Oh, no, he
Sediment.
stopped.
He stopped and turned.
Clever jellyfish. Oh, now we've got the other side.
It's
though.
now
Want to get the lasers off? Go put it.
facing
Yeah.
away from us.
Lasers
Okay.
off.
You can
And
go
it's
into
about
it.
See the
10
underside of that bell?
centimeters in diameter, and you can see the underside of the jelly.
Mhm. See, it's oh, isn't that beautiful?
It really is.
Yeah, you could get a nice view when we saw the underside of its mouth
Mhm.
area.
You sure could.
Right in the center. Beautiful.
I have you at 7-0 in my
Okay.
camera pilot.
About 10 more seconds to this video. And then
Roger.
I'll move on. Actually, we can hold it a little longer.
Yeah, if you
It's
have
really nice.
a minute
It's okay.
if you
10
want.
seconds is perfect. And clear.
Right.
Thank you, Pilot.
Yeah. It's a little hard to get a.
Thank
read
you,
on the
pilot. That
numbers.
was really nice video.
Very
It's okay,
nice
it was
view.
nice that he turned
Thank
over.
you.
Yeah. Okay.
So Gene Ranke confirmed, yes. So ichnology is defined as organism-environment interactions and usually as preserved in sedimentary structures. And neoichnology is the study of extant systems. So systems that are still existing, haven't been fossilized yet. Okay. Well, more stuff in the water column here now.
Okay, let's get a.
What's that? Let's see what that
Get a zoom on
is.
Roger. Okay to zoom.
Hollow 30.
Oh, wow.
Oh.
It is. Look at that.
What
It looks to me like a
Yep.
teeny
It is.
tiny
It is.
holothurian.
We learned earlier in this leg that they don't typically swim around and get things from the water column, but they will.
They can swim.
But they can push themselves off of the bottom and get suspended in the water column, use their modified tentacles to float around or swim around.
Oh, they can propel themselves
yeah.
pretty nicely.
And then they'll go and land on another area of the bottom. This one's been eating for a while, so you can see its
its gut is
packed full of sediment.
well, not entirely full, but it certainly has been chewing on mud.
It's getting awfully close.
Okay, so Bob Carney from Louisiana State says this is Scotoanassa or
Down.
Peniagoni, and
Let's
it
see,
could
come with the
be, it is a, he says it's a pelagic holothurian.
How would it get that much sediment out of the ocean water? It's quite beautiful when backed by the blue ocean.
Oh,
you
it
can
is.
see more of the
It
structure
is.
of the animal.
Oh my gosh.
And the head is to the upper right. The other end is to the lower left.
That's about 8-0. in
They
my
do have a
camera,
mantle
so
that
just,
they
one more
use to
minute.
propel themselves through the water.
Getting too fast.
Yeah.
Thank you.
Around
yeah,
the
head.
that's nice.
Is that wide?
That's full wide, yep.
It's kind of an awesome photo. Sediment in the background.
Get you out of here. There we go.
So Bob says these guys
Try
will
one more?
drop
Sure.
to the bottom to feed, but
Okay.
they do swim through the water column
There we
as
go.
we're watching. And now it's moving a lot faster.
That might have been me.
Doesn't like to be too close to the ROV.
Okay, let's go wide.
Swirling around in the
Roger.
There's
Very wide.
prop wash
Yeah.
from
Yeah.
our propellers will kick up a current.
So Gene Ranke is asking, he's curious, is the fecal matter all organic or is there some calcium carbonate in there or other minerals?
Okay, now I'll move co-pilot.
Yes.
Copy that.
Yes, what? Is it all organic or are there minerals in it?
Oh, sure, there can be minerals in it.
Absolutely.
Nav, when did that move end?
A
We've been
lot
stopped
of
for 10
the
minutes now.
debris
Okay.
that settles out on the seafloor in this part of the Mariana
Yeah,
system
pilots, if you want to push
is
out, then
derived not
probably
just
another
from
move in.
pelagic
All right.
fall or
Uh,
things in the
maybe.
water column that fall to the seafloor,
Gonna
but
be a little
because the
ahead.
volcanic
Yeah.
arc is very active and explosive volcanism occurs there, some of the sediment that I took a look at, scraped off some of the rock samples we've collected on this cruise, contain little broken fragments of olivine, which is a green peridote mineral. There are fragments of broken black volcanic glass. There are cleavage fragments of plagioclase feldspar, which is the most abundant mineral on the surface of the Earth. There are fragments of cleavage fragments of pyroxenes as well. Occasionally long, skinny, sort of prismatic, triangular